self-introduction. I was, let me just take one more step here. I was born in London in the United Kingdom, and I spent my childhood living in England, Australia, and Canada. My undergraduate training was in applied mathematics and physics, and I had every intention of becoming a mathematical physicist. However, during my summer research internship between junior and senior years, I was asked by my nuclear physicist supervisor to do some programming for a collaboration he joined to help plant scientists visualize flexible rod-shaped RNA viruses using electron microscopy. By the end of that summer, I, if you'll excuse the pun, had been bitten by the biology bug, and I decided to pursue graduate training in structural biology. Halfway through my DPhil at Oxford, I had another epiphany and realized that I didn't know nearly enough biology to be useful as a physicist applying quantitative methods to studies of biological systems. After some soul searching, I chose to solve this problem by coming to the United States after graduating from Oxford and taking what could be viewed by some as a step back to go to medical school. The next 8 years of med school, followed by internship and residency training in internal medicine, were a blur of learning human biology and doing postdoctoral research in protein crystallography at night and on the weekends. I didn't sleep very much in the 1980s. After Boston, I went to the Rockefeller University in New York City to start my independent laboratory as a Howard Hughes Medical Institute investigator. I spent 12 very happy and productive years at Rockefeller studying the structural biology of gene expression. But towards the end of my stay there, I felt that I'd stopped learning and realized that I needed to find a new set of challenges. So in 2002, I decided to go over to the dark side to lead R&D at a newly formed biotech company in San Diego, focused on structure-guided discovery of anti-cancer drugs. In 2008, we were fortunate enough to be able to take SGX Pharmaceuticals, Inc. public on the NASDAQ, and a little bit later, we sold the company to Eli Lilly and Company, based in Indianapolis, Indiana. I then spent 4 very interesting years as a Lilly executive before attempting retirement in 2012. And the fact that I stand here today speaking in the Institute seminar series is proof that my retirement was a dismal failure. Fortunately, Rutgers and 2 very fine gentlemen, Ken Breslauer and Chris Malloy, were there to pick up the pieces, if you will. It's an honor to present today's IQB seminar on behalf of the RCSB Protein Data Bank. My talk is rooted in the fact that function follows form in biology. Put less succinctly, the atomic level 3-dimensional structure of a biological macromolecule dictates its function. The importance of looking at atomic level structures of biomolecules in 3D became immediately apparent when the field of structural biology was born in 1953 with elucidation of the double helix structure of B-form DNA by Watson and Crick. They went on to share the 1962 Nobel Prize in Physiology or Medicine with Maurice Wilkins for this discovery. By virtue of GC and AT base pairing, both DNA strands contain exactly the same genetic information, allowing them to be separated to generate identical daughter DNA molecules during cell division. The value of a direct look was equally apparent when the X-ray crystal structure of deoxyhemoglobin S was determined about 2 decades later by Warner Love and his colleagues. They revealed how the glutamic acid to valine change at position number 6 in the beta chain promotes aggregation of hemoglobin tetramers into sickle fibers. Without further ado, I'm going to tell you about exploring experimental structures and computed structure models from artificial intelligence, machine learning at the RCSB Protein Data Bank, rcsb.org web portal. I'll describe some implications for basic and applied biomedical research. A brief outline of my presentation is provided on this slide. I'll introduce the Protein Data Bank and the RCSB PDB, which is probably Rutgers' best kept secret. I'll then move on to a discussion of the impact of the PDB on basic and applied oncology research, US FDA new drug approvals, and protein structure prediction. Thereafter, I'll devote the rest of my talk to explaining how we've integrated computed structure models with PDB structures on our rcsb.org research-focused web portal, and then spend some time presenting vignettes that highlight the impact of both types of 3D biostructure data on basic and applied biomedical research. The Protein Data Bank, or PDB, as I'll refer to, was established as the first open access digital data resource in all of biology. We have just entered our 52nd year of continuous operations, having been founded in 1971 with just 7 X-ray structures of proteins. Some of the structures that motivated protein crystallographers to establish the PDB are illustrated on the right. They include myoglobin and hemoglobin, the first 2 structures to be determined, earning Sir John Kendrew and Max Perutz the 1962 Nobel Prize in Chemistry. The other 2 structures in the lower right panel of ribonuclease and lysozyme were among the first enzyme structures to be determined using X-ray crystallography. Since its inception, the archive has grown more than 24,000-fold to become the single global resource for experimentally determined atomic-level 3D biostructure information. The PDB now holds nearly 200,000 atomic-level structures of proteins, nucleic acids, viruses, and macromolecular machines. Recognizing the global importance of the Protein Data Bank, it has been managed since 2003 by the Worldwide Protein Data Bank Partnership, including PDB data centers in the US, Europe, and Asia, and 2 specialist data resources for electron microscopy and NMR spectroscopy. PDB data are distributed under the most permissive Creative Commons CC0 license at no charge and with no limitations on usage. I'm not exaggerating when I tell you today that the Protein Data Bank served as a vanguard of the open access movement, exemplifying the FAIR principles of findability, accessibility, interoperability, and reusability long before they were first articulated in print in 2016. The RCSB Protein Data Bank is the US data center for the Worldwide Protein Data Bank. We convert global data into global knowledge. The organization is headquartered here at Rutgers within the Institute for Quantitative Biomedicine, located in the Proteomics building on the Bush Science campus here in Piscataway. 2 smaller RCSB PDB teams are based at the University of California, San Diego, and the University of California, San Francisco. Day-to-day operations are supported by 4 interlocking services, including 1, data deposition and biocuration, which is carried out in concert with our WWPDB partners located in Europe and Asia. 2, archive management and access, reflecting our role as the Worldwide Protein Data Bank designated archive keeper. 3, data exploration via our rcsb.org research-focused web portal, which is visited annually by many millions of PDB users based in virtually every sovereign country recognized by the United Nations. And 4, training, outreach, and education via our pdb101.rcsb.org website. PDB data are also repackaged and redistributed by more than 450 external data resources and used intensively within biopharmaceutical companies, both large and small. 2 publications listed at the bottom of this slide describe the activities of the RCSB PDB. We're jointly funded by the National Science Foundation, the National Institutes of Health, and the US Department of Energy. Federal funding of the PDB for more than 5 decades is a strong testament to the value of 3D biostructure data and RCSB PDB services for basic and applied researchers, educators, and students across fundamental biology, meaning the NSF, biomedicine, the NIH, and energy sciences, the Department of Energy. 4 recent publications documenting this breadth of impact are listed at the bottom of this slide. PDB holdings have had a significant impact on basic and applied cancer research. We're all, I think, well aware that RAS genes are mutated in approximately 1/3 of all human cancers. RCSB-PDB supports the National Cancer Institute RAS initiative with an extensive data ecosystem of RAS and RAS-related structures. The PDB also facilitates drug discovery targeting RAS, and drug discovery targeting RAS interacting proteins, such as the son of Sevenless or SOS depicted on this slide. Finally, protein data bank holdings provide opportunities for understanding target biology in 3D and initiating structure-guided drug discovery for signaling proteins downstream of RAS. The various numerical values here provide you with a sense of how extensive PDB holdings are for each of these different actors in the RAS signaling pathway. Excuse me. Let me just go back one. Having provided you with this overview, I'm going to present a series of vignettes beginning with the impact of PDB data on US FDA new drug approvals. Thus far, we've published 3 papers on this topic. First, we analyzed approvals across all therapeutic areas for the 7-year period 2010 through 2016. This graphical abstract summarizes findings that our late colleague John Westbrook and I reported in 2019. Discovery and development of 184, nearly 90% of the 210 new drugs approved by US FDA from 2010 to 2016 were facilitated by open access to nearly 6,000 PDB structures. Impact of 3D biostructure data ranged from better understanding target biology all the way to structure-guided drug discovery. I'll return to the vemurafenib BRAF example depicted on the right-hand side of this slide shortly. A better understanding of target biology may not sound like particularly important, but having spent a decade working in the biopharmaceutical industry, I can assure you that it is. An influential study published by Kohler and Landis in Nature Reviews Drug Discovery documented that approximately 30% of attrition in drug discovery and development across the biopharmaceutical industry can be attributed to so-called efficacy failures. By that, I mean that the would-be drug interacted with the intended target biomolecule and had the desired biochemical or biological effect, but the expected clinical benefit was not realized. Because efficacy failures typically occur during phase 3 clinical trials, they can be extremely expensive for the sponsor company and are best avoided by understanding target biology as well as possible. An independent analysis of the same 210 drug approvals was published by Galkina Cleary et al. In the Proceedings of the National Academy of Sciences. The citation is provided at the bottom. Their work, interestingly, revealed that NIH funding of pre-competitive research focused on the 150 or so protein targets of these 210 newly approved drugs between 2010 and 2016 totaled around $100 billion. Our work and that of Galkina Cleary et al. Underscore the importance of public funding of basic research and open access to data underpinning the global drug discovery and development ecosystem. $100 billion in NIH funding and open access to nearly 6,000 PDB structures ultimately attracted more than $600 billion in biopharmaceutical company investment to bring the 210 new drugs to the market. In my opinion, at least, a 6-to-1 multiplier resulting in drugs that will improve the lives of millions of patients worldwide represents an entirely appropriate use of US taxpayer dollars. Our second paper on this topic, published as a cover article in Drug Discovery Today, examined approvals of anti-neoplastic agents over 9 years from the beginning of 2010 to the end of 2018. This graphical abstract summarizes findings we reported in 2020. Discovery and development of 74, that is around 95% of the 79 new anti-cancer drugs approved by US FDA 2010 through 2018, were facilitated by open access to nearly 2,500 PDB structures. 54 of the new drugs were small molecules, meaning they administered as pills, and 25 were biologics, administered by injection. More than 70% of the 54 newly approved small molecule anti-cancer drugs were the products of structure-guided drug discovery supported by protein crystallographers working for the biopharmaceutical companies. In every case, these structural biologists would have been able to begin their work with a PDB structure of the target protein or one very closely related in amino acid sequence and 3D structure. For the 25 newly approved biologics, company researchers would have been able to use PDB structures of antibodies and cytokines and cytokine receptors to guide molecular design decisions. Arguably, the best known of the 25 biologics approved between 2010 and 2018 were the two anti-PD-1 antibodies, pembrolizumab from Merck and nivolumab from Bristol-Myers Squibb. James Allison at the MD Anderson Cancer Center and Tasuku Honjo at Kyoto University shared the 2018 Nobel Prize in Physiology or Medicine for their discovery of cancer therapy by inhibition of negative immune regulation. Honjo is credited with discovery of PD-1, the target of pembrolizumab and nivolumab. For those of you who'd like to learn more about the impact of 3D structures on small molecule drug discovery and development, please consult a review article that I published last year in a special issue of the Journal of Biological Chemistry, celebrating the 50th anniversary of the founding of the Protein Data Bank. Returning to the example illustrated 4 slides earlier, the vemurafenib origin story persuasively demonstrates the importance of open access to PDB data and the power of structure-guided drug discovery. The first BRAF protein kinase structure was deposited to the PDB in 2004 by a UK academic group. Their work documented that some of the previously known cancer-causing mutations mapped to the catalytic domain of BRAF depicted in the right-hand panel. In 2007, it became clear that valine 600 to glutamic acid, or the V600E somatic mutation, is the driver in approximately 50% of advanced stage melanomas. Plexicon, a Bay Area biotechnology company, used fragment-based structure-guided drug discovery to discover and develop vemurafenib, which is a highly selective inhibitor of V600E BRAF, but importantly, not the wild-type enzyme. They published the co-crystal structure of BRAF bound to vemurafenib in 2010 and deposited the atomic coordinates to the Protein Data Bank. Their decision to make the atomic coordinates public was not entirely altruistic. PDB data deposition is mandatory for publication of new 3D biostructures in all major scientific journals. Vemurafenib was subsequently approved by US FDA in 2011 for treatment of late-stage melanomas bearing the V600E somatic mutation confirmed by an approved genetic test. Not unexpectedly, acquired resistance has limited its utility as a single-agent anti-cancer treatment. One of the common resistance mechanisms involves upregulation of mitogen-activated protein kinase, or MEK, activity. Hence, vemurafenib is now being used in combination with MEK inhibitors such as cobimetinib. Plexicon was the lone innovator when they discovered and developed vemurafenib targeting V600E BRAF. The following vignette focused on CDK4, CDK6 inhibitor discovery is more typical of new drug approvals in the biopharmaceutical industry. Both CDK4 and CDK6 represent important targets for treatment of breast cancer. The first PDB structure of the catalytic domain of CDK6, shown here on the right, was deposited to the Protein Data Bank by Nikola Pavletic and colleagues in 1998. It was studied in a binary complex with multiple tumor suppressor. The first PDB structure of the catalytic domain of CDK4, shown here on the right in purple, was deposited to the Protein Data Bank in 2009. It was studied in a binary complex with cyclin D1, shown here in green. With the benefit of these and other pre-competitive structures archived in the PDB, 3, possibly more, major biopharmaceutical companies went head-to-head to discover orally bioavailable dual CDK4, CDK6 inhibitors using structure-guided approaches. In 2017, 3 companies, including Pfizer, Novartis, and Lilly, received US FDA new drug approvals for their dual CDK4, CDK6 inhibitors. I'm certain that the protein crystallographers working at all 3 companies use the many pre-competitive PDB structures of CDK4 and CDK6 as starting points for their efforts. I witnessed the very thing when I was a Lilly executive for those 4 years. In 2016, Pfizer generously published co-crystal structures of CDK6 bound to each of the new drugs approved the following year and deposited them to the protein data bank. All 3 compounds are chemically distinct from one another, yet each compound binds in the hinge region of CDK6 between the 2 lobes of the kinase domain, making comparable hydrogen bonds to the polypeptide chain backbone. They do, however, make distinct hydrogen bonds and van der Waals interactions with other parts of the enzyme active site. As distinct chemical entities, they are very likely to have different side effect profiles, which could benefit women with breast cancer who may find that they can tolerate one drug, but perhaps not the other 2. Let me turn now to another contribution of open access to PDB data, and that is the emergence of computational structural biology as a vibrant scientific discipline. Inspired by the work of Anfinsen, who showed that the sequence of a polypeptide chain determines its shape or fold, computational biologists tackled the challenge of predicting the 3D structure from sequence alone. Initially, they succeeded with homology or comparative protein structure modeling using closely related templates from the PDB. Homology modeling works because of the parsimony of evolution. Generally accepted estimates put the total number of distinct protein domain folds or shapes in nature at about 10,000. It appears that once nature has found a protein sequence fold combination that works, it is reused many times to support similar biochemical or biological functions. As the number of structures in the PDB grew, computational biologists began learning how to predict the structures of small proteins without the need for templates. In 2020, Google DeepMind announced that their artificial intelligence machine learning AlphaFold 2 software can predict the structures of large proteins with accuracies comparable to low-resolution experimental methods. Their approach relied critically on open access to PDB data and genomic sequence evolutionary covariance information. This was a landmark achievement in structural biology. The first author of the AlphaFold 2 Nature paper, John Jumper, and the CEO of Google DeepMind recently shared one of the breakthrough prizes and about $3.5 million. The Rosetta team, led by David Baker, a Howard Hughes Medical Institute investigator at the University of Washington, quickly produced a rival software tool known as Rosetta Fold, and subsequently Rosetta Fold 2, which has similar performance to that of AlphaFold 2. David previously received a breakthrough prize for his pioneering work with Rosetta using evolutionary covariance, which set the stage for successful application of artificial intelligence machine learning methods to the protein structure prediction problem. Much happened very quickly since AlphaFold 2 was announced in late 2020. Reasonable accuracy computed structure models, or CSMs, are now freely accessible for nearly every protein sequence represented in UniProt. This graphic from our 2021 New England Journal of Medicine paper, co-authored with Professors Renata Pasqualini and Wadi Arap of the New Jersey Medical School and the Cancer Institute of New Jersey, contrasts experimental structure determination with computed structure modeling. Both workflows rely critically on open access to genomic and 3D structure data. Key differences are as follows. Experimentally determined structures stored in the protein data bank are highly accurate and frequently include bound small molecule ligands, such as enzyme cofactors or drugs, or bound macromolecules, including other proteins, DNA, or RNA, as shown here in this inset. But for human proteins and those from many other eukaryotes, they are frequently limited to individual globular domains for technical reasons. Consequently, protein data bank coverage of the human proteome is only about 20% in terms of the total number of amino acid residues encoded by the human genome. Computed structure models coming from AlphaFold 2 and Rosetta Fold 2 are less accurate on average than PDB structures, and as a rule have no bound small molecules or other macromolecules. But most of these computed structure models cover the entire polypeptide chain, as shown here in this inset. Combining PDB data with computed structure models increases confident structural coverage of the human proteome to nearly 60%. I'll come back to this point later in my talk to explain why confident structural coverage of the human proteome will never reach 100%. For the avoidance of doubt, computed structure models, or CSMs, are not stored in the protein data bank. Many of them are now available open access from the Model Archive, coming from diverse contributors, and from AlphaFold DB, coming solely from Google DeepMind. At the end of August 2022, the RCSB PDB team released an updated version of our rcsb.org research-focused web portal that now provides a one-stop shop for 3D biostructure data. This graphical abstract summarizes current RCSB PDB offerings. We deliver nearly 200,000 experimentally determined PDB structures alongside more than 1 million computed structure models coming from the Model Archive and AlphaFold DB. PDB structures and computed structure models are clearly identified as to their provenance and reliability. Both types of 3D biostructure data can be searched, visualized, and analyzed using the full functionality of rcsb.org. I'm going to begin talking about computed structure models in more detail with the topic of model organism structures in the PDB. I told you that structural coverage of the human proteome in the PDB was limited to about 20%. The situation is even worse for model organism proteomes. It's been limited historically by funding and other pressures such as advancement and promotion. For example, only about 4% of Arabidopsis thaliana proteins are actually represented in the PDB. Simply put, it's easier to get an NIH grant to study the 3D structure of a human protein implicated in disease than its plant ortholog. And studying the human proteome, the human protein may make it easier to be promoted or receive tenure in a university. Regrettably, the situation is similar for other model organisms, ranging all the way up to mouse and rat. PDB coverage of the mouse proteome is very limited. By integrating more than 1 million computed structure models into rcsb.org, we're now delivering 3D biostructure data from most model organisms, selected pathogens, and organisms important in bioenergy. The example shown here on the right highlights the fact that the yeast protein structure, PDB ID 1 Bravo 54, using the NATO alphabet, has 15 homologs or orthologs in the PDB, all coming from either yeast or bacteria, including more than 1 million computed structure models alongside the PDB structures, has increased the number of homologs or orthologs of PDB ID 1 Bravo 54 available from rcsb.org to 81, including mouse, human, fly, worm, etc. Providing a one-stop shop access to both PDB structures and computed structure models is going to allow us to better serve the 99% of rcsb.org users who are not experts in structural biology and will probably never determine the 3D structure of a macromolecule. They are often frustrated. They were often frustrated, I should say, in the past because their favorite protein was not represented in the PDB. While it's true that many computed structure models of globular proteins recapitulate what was already known from the protein data bank, there's no doubt that AlphaFold 2 and RosettaFold 2 can correctly predict previously uncharacterized structures. This slide shows the AlphaFold 2 computed structure model of full-length human Tweety homolog 2. This is a protein implicated in cancers and other diseases, including epilepsy, chronic pain, and viral infections. Most of the CSM that you can see here on the right-hand side of the slide is color-coded blue or cyan to denote confidently predicted atomic coordinates. The C-terminal 130 residues, however, are color-coded yellow and orange because their atomic coordinates were not confidently predicted by AlphaFold 2 and should not be trusted. And here's the electron microscopy structure of human Tweety homolog 2 from the PDB, published after Google DeepMind made the prediction shown on the previous slide. It's a symmetric, homodimeric, integral membrane protein. The location of the lipid bilayer is denoted by these two gray discs, and the sites of 3 glycosylation sites per protemer are shown here, here, and here. The C-terminal 130 residues were not visible in the electron microscope. They're almost certainly intrinsically disordered, meaning they lack a defined 3D structure. Hence, they could not be experimentally visualized or predicted confidently by AlphaFold 2. The confidently predicted portions of the AlphaFold 2 computed structure model are quite good. The backbone atom positions differ on average by about 0.8 angstrom units from the ground truth of the PDB experimental structure 7 Peter 54. For reference, the diameter of the hydrogen atom is about 1 angstrom unit. When I was a Howard Hughes Medical Institute investigator at the Rockefeller University, I had the good fortune to collaborate with Professor Robert Darnell and his colleagues on structural studies of human Nova-2 protein, which is a nuclear RNA binding protein implicated in various neurodevelopmental disorders. We were unable to crystallize full-length Nova-2 despite considerable efforts, but we did succeed in determining 3D structures of the third of 3 K homology or KH domains, both with and without RNA bound. A ribbon diagram of the Nova-2 KH3 protein RNA complex is shown on the right with the protein in purple and the RNA hairpin in orange. The AlphaFold DB computed structure model of full-length Nova-2 is shown on the right. It reveals confidently predicted structures of 3 KH domains as expected, but about 70% of the protein could not be predicted reliably by AlphaFold 2. The yellow and orange segments of the polypeptide chain are almost certainly disordered. Nova-2 is a typical beads on a string eukaryotic protein in which the beads are globular domains and the string is the disordered regions. This observation explains 1, why we were never able to crystallize full-length Nova-2. Parts of the protein are simply too flexible, and 2, why integration of computed structure models with PDB data did not increase coverage of the human proteome beyond about 60% of amino acids encoded by the human genome. Most of the remaining 40% are almost certainly disordered, lacking defined 3D structure. Combining information from PDB and computed structure models can enrich the RCSB.org customer experience. On the right, I've overlaid the PDB structure of Nova-2 KH3 bound to the RNA hairpin in gray onto the Nova-2 computed structure model to present our knowledge of RNA recognition in the context of the full-length protein. I think you can readily appreciate that the other 2 KH domains, which have different RNA binding specificity, could simultaneously bind to another RNA molecule or a different segment of the same RNA chain. This type of analysis can now be carried out using our research-focused RCSB.org web portal. More information about the new capabilities of RCSB.org will soon be available in a nucleic acids research publication that's just been returned to the journal and is about to enter production. As of Sunday night, the PDB contained more than 1,100 experimentally determined 3D structures of the SARS-CoV-2 spike protein, including variants and those with bound FABs, nanobodies, etc. In an ongoing collaboration led by our colleague, Professor Sagar Kare, we're using computed structure models based on PDB structures of the SARS-CoV-2 spike proteins to assess the likely impact of amino acid changes on antibody binding. This work will shortly be submitted for publication with Joey Lubin, who's credited with this figure as the first author. Thank you, Joey. In a recently concluded collaboration led by Professors Renata Pasqualini and Wadi Arap here at Rutgers, members of the New Jersey Medical School and the Rutgers Cancer Institute of New Jersey, PDB data and computed structure models were used to identify a universal epitope of the SARS-CoV-2 spike protein, denoted cysteine 662 to cysteine 671. This segment of the spike protein with amino acid sequence CDIPI GAGIC is fully conserved across all observed major variants of SARS-CoV-2 and SARS-CoV. This work was published in a molecular biology and evolution paper with New Jersey Medical School MD PhD student and former Rutgers MBB student Christopher Markosian as the first author. The invariant decapeptide epitope represents an attractive candidate for use in a phage-based SARS-CoV-2 vaccination strategy designed by Professors Pasqualini and Arap and published last year with Professor Daniel Stacquicini, postdoctoral fellow Fenny Tang, and again MD PhD student Markosian, designated as co-first authors. In another Rutgers collaboration led by Professor Stephen Zheng at CINJ, we used PDB structures and computed structure models to investigate the impact of mutations within the active site of mTOR. This work sought to understand the potential impact of mutations that could arise during targeted inhibitor therapy. Stephen and his colleagues showed experimentally that, contrary to expectations based on earlier observations with BCR-ABL, mutation of the gatekeeper residue, isoleucine 2237, does not affect enzyme activity or inhibitor binding. Within the active site, the mutational hotspot of mTOR is leucine 2185. The L2185 alanine mutation still works as a protein kinase, but confers resistance to a variety of inhibitors, including PP242 and sapanisertib. This mutation, however, is still sensitive to inhibition by taurin2 and dactylisib. Computed structure models based on PDB structures were used to explain the observed patterns of inhibitor resistance and sensitivity, providing a framework for understanding mTOR inhibitor resistance when it begins to emerge in the clinic. This work was published in Cell Reports. In another Rutgers collaboration led by Professor Sridhar Ganesan at the Cancer Institute of New Jersey, my RCSB PDB colleague Brinda Vallat and I used PDB structures and computed structure models to investigate a novel combination of somatic mutations occurring in epidermal growth factor receptor, or EGFR. These mutations were identified in the peripheral blood of a patient with non-small cell lung cancer who experienced disease progression on osimertinib. Computed structure models of the double mutant with various bound drugs explained: 1, kinase activation by the leucine 858 arginine somatic mutation, and osimertinib resistance due to the methionine 766 to glutamine somatic mutation. They also allowed us to predict that protein kinase activity would be inhibited both by neratinib and poziotinib, which was later confirmed by Sharon Pine and her colleagues in various model systems. This work was published in the Journal of Thoracic Oncology and accompanied by an expert commentary co-authored by Doctors Klempner and Hatta entitled Sequence, Treat, Repeat. The paradigm that they're referring to here is that one does genomic characterization of the tumor DNA, identifies a potential targeted therapy. You begin treating the patient with that targeted therapy with the goal of managing the disease for the longest extent possible. But when there is an emergence of resistance, when the treatment begins to fail, you repeat the process of sequencing, identifying what the new driver mutations are, because they're not those that were originally present when the tumor was first sequenced. Select a new treatment with the goal, again, of managing the cancer for as long as possible. So as I begin my close, I'm optimistic that I've made a convincing case for the fact that reliable 3D biostructure data, be it experimental or computed, will enable research and education across biomedicine for many years to come. At the RCSB Protein Data Bank, we're integrating experimental structures with computed structure models and making this corpus of data readily accessible and understandable for basic and applied researchers across fundamental biology, biomedicine, energy sciences, and bioengineering and biotechnology. My take-home messages are as follows. Open access to PDB structure data informs research across the biosciences. Open access to PDB structure data has contributed substantially to US FDA new drug approvals. And open access to PDB structure data was central to recent advances in computational structure prediction using artificial intelligence machine learning methods. Combining PDB structure data with computed structure models is going to accelerate research across the biosciences. The RCSB Protein Data Bank, rcsb.org research-focused web portal, is now a one-stop shop for using 3D biostructure data in your research and teaching. I'm going to close with an open invitation for collaboration with the RCSB Protein Data Bank. Please do not hesitate to contact me via email at stephen.burley@rcsb.org if you're interested in using 3D biostructure data in your research or teaching. For an introduction to PDB data, please see pdb101.rcsb.org. I thank you in advance and wish you health and safety. And in closing, I thank you for your attention. I need to acknowledge the large team of dedicated professionals working at the RCSB PDB shown in this pandemic view. Their efforts allow me to stand at the tip of the spear and present to you today. I thank our funders of the National Science Foundation, the National Institutes of Health, and the US Department of Energy. I thank Rutgers, UCSD, and UCSF for hosting the RCSB PDB, and I thank my Worldwide Protein Data Bank colleagues in Europe, Japan, China, and Biomag ResBank and Electron Microscopy Data Bank for their partnership. Finally, I have to acknowledge the more than 50,000 structural biologists who freely contributed nearly 200,000 structures to the PDB, and the many millions of PDB data consumers from around the world who are using 3D biostructure data to drive advances in research and education that are changing the world. In the time remaining, I'll be happy to entertain questions, any and all. Thank you. So if you would like to put any questions in the chat, I'll be happy to answer them. But Vivian, I see you've raised your hand, and I'm going to allow you to talk. Thank you.
Thank you.
Good to see you.
You too, Stephen. The unstructured regions of proteins, they're unstructured because they kinetically move, right?
They're either statically or dynamically disordered, yes.
Okay. If they're dynamically disordered, does that mean there's ever a time when we'll be able to understand where they are when they're, at a certain pH or bound to a certain substrate, or just like we can look at structured areas and see how they move when they're bound to a substrate? will we ever get any further in understanding the unstructured regions? How do you envision?
So when I was at Rockefeller, I did some work in this area, and it's clear from work that we did and work that others have done, and Jane Dyson at Scripps has described in some review articles, some of these disordered segments of proteins undergo disorder to order transitions when they bind to their target protein, when they actually find their interaction partner. And we showed that with both the EIF4E binding proteins and with EIF4E, when they interact with the cap binding protein, EIF4G. Those of you at Rutgers will remember that the mRNA cap was actually discovered here at what became CABM. So in that case, it's a completely disordered protein that adopts an alpha helical structure when it binds to the dorsal surface of EIF4E, the anterior surface being the site of the cap binding So it is definitely possible to visualize the consequence of these disorder to order transitions in selected cases, and that's not the only one. Jane Dyson, I think, has done a very nice job in providing reviews of some cases that are informative. And we do know from analyses that we've done across the PDB comparing, this is work of Cheng-Hua Hsiao that was just published in Structure in the most recent issue. We do know that there are segments of polypeptide chains that could not be predicted confidently by AlphaFold 2 that actually have defined 3D structures in the PDB. Because they're in contact with another target protein.
Thanks.
But my message to the 99% of RCSB.org users who are not structural biologists, who are never going to determine a structure, is that the 40% of the human proteome for which we don't have confident structural information is the place where you should be planning experiments that use techniques other than 3D structure determination to understand what's going on.
Okay.
So thank you, Vivian. Shantanu Jha has asked a question. Good to see you online, Shantanu. Thank you for joining. Would RCSB users find it useful to include the ability to take a computed structure model and refine it further using computational capabilities? And then Shantanu goes on to ask, would RCSB consider linking to such computational workflow capabilities via NSF or DOE supercomputers or clouds? We're certainly open to acting as a portal for individuals who are seeking to access other data resources. And in fact, there's quite a number of places on the rcsb.org website where we provide links to external data resources. So we're certainly open to that. I'd be happy to discuss it with you, Shantanu. Well, if there are no further questions, I thank you for your attention. And I think we should draw this to a close because I've got to take a breather before I meet with the IQB graduate students in a few minutes on another Zoom link. So thank you very much for participating today. And don't forget that our next seminar to be given by Andras Fieser, hosted by Brynda Vallat, will take place at noon on November the 9th, on Wednesday, November the 9th. And I'm very confident that Andras is going to give a great talk. Be safe, everybody.